That's me on my own at the moment, Billy, but that's all right.
Can you hear me, Milton?
I can hear you very clearly.
And can you still see me?
I can see you.
Okay, I can't see myself. That's good. What about the box?
I'll get rid of that in a minute. This meeting is being recorded box.
Oh no, not that one. The one where so you can see my screen, right? But the one where everyone, so the screen where I can see you, is that coming up on your end?
It is coming up on our end, yeah.
Yeah, okay.
You probably want to get rid of that.
Yeah,
so if you go to that.
Yeah.
I think if you click on that one.
Yeah, I can just put this down the bottom here. I'm in this work, so I won't get nervous by other people watching me. This is perfect. Okay.
No, you're right.
Yeah, it's fixed.
Not on this end, it's not that.
Isn't it?
On this end, I've still got this meeting is being recorded box.
Oh no, not that box. I meant the box where you can see the seminar room and everyone who's present.
I've still got that on my screen.
Oh, okay.
Which I think I'd have to do that to get rid of it. And then
yeah, it might be in the way of the slides at some point. That's all. That's what I was thinking.
Well, no, now I got rid of it for you, Billy.
Okay, perfect.
So
thanks, Milton.
Well, I have no idea what I'm doing. So. I'm relying on others with far more expertise than me, Billy. That's how it works. I'd say because the seminar, the normal seminar host has just arrived, so if I get to start.
I'm sure it should be fine. Like you said, we'll fumble our way through it.
Oh, we'll fumble our way through, but it'll be right.
Yeah.
I'm just counting how many seats.
Did you notice too that the bar where the Q doesn't work with the G scanning system? That's pretty annoying, isn't it? Because I like the Queensland scanning thing because you don't have to keep on. I'm sure those Melburnians have got it worked out by now, haven't they, Billy?
Worked out with what? Sorry, I couldn't quite hear.
The scanning thing, you know, when you go to
oh
places,
yeah. The barcode. Yeah. Well, Android phones won't let you scan by default, so you have to download a different app. It took me about 2 months of lockdown to figure that out.
Okay.
I was always asking for paper and annoying the restaurant staff, saying my phone couldn't do it.
We'll be careful what we say here, Billy.
Okay, sure. No,
about lockdown and things like that. Okay.
Yeah.
Zoom is listening.
Yeah, we've definitely got it sorted out down here.
We'll just wait a couple more minutes for the stragglers to wander in. Teaching is online. You don't have lectures. Lectures are online. No labs are in. Oh, just in that. And some shoots, if you really like it, not a great world. But even I don't know. Really not worth it. And if I sat in the class, but really, do I need to repeat? I can't work any class. I didn't expect those lines. Remember how it's really how they show. Relationship way around that. So I guarantee on the 50%.
All right, should we start? We probably should. Are you there, Billy?
Yeah, I'm here.
All right,
Ready to
excellent.
start.
I'm
when
going
you
to
are.
I'll give a brief intro to people, everyone. I'm going to stand over here, so I should explain. We had a bit of a technical glitch in that the lapel mic won't work on my laptop because apparently my laptop's running an operating system that disappeared in the time of the dinosaurs. Nonetheless, at least Zoom's working, so that's good. So I'm just going to stand here and chat. So for those who don't know, so today we're fortunate to have Billy Williams Noonan give us a presentation. So a bit of background about Billy. Sorry to embarrass you, Billy. I first met Billy when he was in year 10 at high school, and he came to Glycomics and did work experience in my lab. So that's when I first got to meet Billy. I won't say how many years ago that was, Billy, because I've forgotten. But that's all right.
Who knows?
A little while. Anyway, so that's when I first met Billy, and then Billy came to Griffith, did his Bachelor of Science degree. He did his honours degree with me in the Chem lab. And then he took a bit of a break, and then I encouraged Billy to explore a PhD where he could put his computational skills to work. And so Billy then went and did a PhD at, I always call it Pharmacy College, but MIPS, so Monash Institute of Pharmaceutical Science, with Dave Chalmers as supervisor. And part of his talk today, the first couple of topics of his talk today are lifted directly out of his PhD, which I think he finished, well, I'll say a bit over a year ago, Billy. It's probably a little bit longer than that now.
It was. I got awarded the title in 2020, so you're right.
Excellent. All right, thanks, Billy. And so then Billy now is a postdoc at RMIT, where he's doing some of his SARS-CoV-2 research that he's going to talk about today. So I'm going to hand over to Billy, and his talk is computational free energy methods to assist drug design efforts in targeting HIV-1 integrase, influenza virus into neuraminidase, and SARS-CoV-2. So Billy, it's all yours.
Thank you so much, Milton, for having me here for, today, and to Daniel and Sarah, everyone, for helping to organise this. You've been really gracious. It's a shame I can't be here in person, but I'm excited to be presenting this work all the same, and I'm glad you've come. So today we're talking about the capacity of computers to predict key pharmaceutical properties for 3 specific retroviruses or 3 specific drug targets. These drug targets are HIV-1 integrase, influenza virus N2 neuraminidase, which I'm sure everyone here knows a wealth of information about. And the third drug target, or the third mechanism that we're looking at, is SARS-CoV-2 S protein adhesion. So before I get into those 3 specific case studies that we're going to talk about today, I need to give you a little bit of background about computers and what we can reasonably expect from them, and what sort of accuracy we can expect in predicting things like KD, the dissociation constant for protein ligand binding. So things have certainly changed since the 80s and 90s, and even since the early 2000s with the advent of GPUs, for example. Things have changed in the last 50 years. Wait, no, it's 40 years, isn't it? But things have certainly also changed since the 1800s. So I'm not sure if you can see this. Yeah, I just moved my thing out of the way. But there was a philosopher, a well-renowned philosopher from France, and I might butcher his name, but his name was Auguste Comte. And he quite famously stated that if mathematical analysis should ever hold a prominent place in chemistry, an aberration that is happily almost impossible, it would occasion a rapid and widespread degeneration of that science. So we now have computers. So that's an example of how technology changing in a way you couldn't foresee has changed how we do chemistry, because we certainly can make calculations of pKa now and predictions that are quite accurate in a way that you couldn't in the 1800s. Things have changed since the 1800s and even since the 80s, as I said. So a computer used to look like the picture on the left in the 80s, and the sort of typical molecular dynamics study that you could expect to be fairly reasonable quality was something like this reference here, where they did molecular dynamics simulations to study the atomic motions of water. So they'd have a box of water and they'd run a simulation. And of all the bonds in the water stayed what you'd expect, and everything ran as you'd expect. And that was a reasonable study in 1985. 6 years ago, in 2015, scientists at a commercial software company in New York called Schrodinger. So what they did was they made predictions of protein ligand binding affinity and potency to assist drug discovery for hundreds of protein ligand complexes, and they published that in JACS. So they had not just a box of water, as was sort of okay quality in 1985, but these scientists they simulated hundreds of protein ligand complexes in water. So computers are certainly changing, and as a sort of example, this is what a computer looked like in the 1980s. Today, you'd expect a desktop to look like this. So computers have improved and technology has changed. And we can use computers to predict key pharmaceutical properties within a certain range of error. And one of the things we're going to be looking at is benchmarking certain computational methods in predicting binding affinity. But computers can also be used to predict membrane permeability, for example. And something that's not often done is that we'd want to make prospective predictions. So the retrospective case where you know the answer ahead of time has often been done, and it's easier because you just keep doing the modeling with different settings until you get the right answer. The prospective case is often harder because you don't have the right answer a priori. So there's been a trend recently in doing this with computational methods, making prospective predictions of KD. And being able to do this would be useful, because if you can predict KD on a computer reliably without making the compounds and without synthesizing the protein, if you can do that, then it can guide laboratory chemists into making active compounds with less effort. And that would save the cost of drug discovery and expedite the process. But importantly, with modeling, who's heard of XKCD? It's like this comic, I find it funny. We can't extrapolate a modeling that works on one test system to everything. So often some of these methods are circumstance specific or system specific, and you can't extrapolate to different systems in the same way that this person here, so he says you had 0 husbands yesterday, so and then you have 1 today. So if we extrapolate, by the end of the month, you'll have 30 husbands. You can't do this sort of thing. So you need to make sure you don't extrapolate our modeling into places that you wouldn't expect it to work, just because we are using heuristics or approximations along the way. But we do have to kind of ask the question, what sort of accuracy can you expect when you're making predictions of binding affinity, for example, on a computer? Before I get into that, I'll tell you about the methods we're using. So the methods, that there are multiple ways of running computational simulations. I think Yauchi's group at Glycomics, they focus on completely different methods to this. But the method I did my PhD in and that I specialize in is molecular dynamics. And what that requires is you have the coordinates of the atoms in a system from, for example, a crystal structure or a cryo-EM structure, and you apply certain parameters to those atoms, so like bonded potentials and so on. And if you solve for Newton's equations of motion after applying velocities to the atoms and temperature and pressure, if you solve for Newton's equations of motion every time increment or time step, you can resolve for the velocity and the positions of the atoms at each of those time increments. And then if you do this over multiple time steps, you can see things move, like the protein on the right. And this can be used to simulate biochemical events or biochemical phenomena on like high nanosecond to low microsecond timescales. If you have really expensive equipment, like Anton in the US, that's a supercomputer that they have there, you can even still simulate small endosomes on the microsecond timescale. So that's if you have the most high quality specialized equipment. In Australia, we can expect maybe 10, 20, 30 microseconds at most, and that's quite hard, but we can do that. So we can simulate these events feasibly, and that's the sort of thing we can expect. So one thing we can do when we're running these simulations is have separate systems. And in order to predict a binding free energy, or a delta G, so binding free energy of binding, we need to look at two different cases. The case where you have the free ligand and the free protein, and you can sample all the relevant conformations or microstates of this state where you have the free ligand and the free protein. That's one state. The other state is the endpoint, where you have the bound protein ligand complex, and you need to sample all the conformations and all of the energetic states or microstates of this specific bound state. So once you have the energy of the bound state as an average of all the energies of all the different conformations, and once you have the same thing for the free state, by subtraction of those two average energies, you can get the delta G of binding. And mathematically, this delta G of binding is a function of the KD. So it's the gas constant times temperature times the natural log of KD. So just by some simple algebra, you can convert between the two if you wanted to. So as I said before, we sort of have to ask the question, how accurate is this? How reliable is this? So as I said, the scientists at the commercial software company, Schrödinger, who've been publishing several papers in this area in recent years, they've been claiming that you can make accurate predictions of protein ligand binding for energies to within 4.6 kilojoules per mole of experiment. So high quality isothermal titration calorimetry experiments, they themselves vary by up to 2.1 kilojoules per mole if you do repeated experiments. So the argument that's being made is that the accuracy of computers is slowly approaching the accuracy of experiment. And this is often done in the retrospective case. There are some papers that have been published where they've made prospective predictions and they're just marginally less accurate, which you could probably expect. But this is all quite exciting. And as computers and algorithms improve, this kind of prediction should also become more reliable. So for this talk, we hypothesize that we can make some reliable predictions of protein dynamics and protein ligand binding affinities, and that we can make prospective predictions in some cases. And the aims are to do that, to make some retrospective predictions of binding affinities, and to also make prospective predictions where we can, and either validate those predictions or leave the validation for a later date. In my case, during my PhD, the validation was outside the scope of my project personally, but the idea is that we could potentially do this. So as I said, we're going to be talking about three specific cases. The first case is with a drug target called human immunodeficiency 1 virus integrase. So HIV is the leading cause of human immunodeficiency, the symptoms caused by AIDS, for example. So it can cause immunocompromisation by people, but importantly, we're going to use this virus or this viral drug target as a test system. So ligands binding to IN or HIV integrase are valid test systems for these free energy studies because the ligands are very rigid, as we'll get into later. So HIV is a retrovirus. It binds to the host cell. It inserts the retroviral RNA that gets transcribed into proviral DNA. And it's this step here that's important. There's a protein that binds the proviral DNA called integrase, and that translocates the proviral DNA through interactions with LEGF or P75, and it translocates the proviral DNA into the host nucleus for integration. And then obviously after the DNA is integrated, the virus can then replicate, bud, and leave. But if we inhibit this step, that's a valid mechanism for a potential therapeutic for HIV integrase, and we're going to use ligands binding to an allosteric site of HIV integrase as test systems for comparing free energy methods. So, importantly for these sorts of comparisons between different free energy methods that we're going to make, there have been studies that compare 1 or 2 or 3 even at most different free energy methods, but here we're going to compare more than that. And we're going to be using these ligands binding to an allosteric site of HIV integrase. So they look like this, and the little substituents, the R groups, can all look like this. And you can see these are quite rigid. They can't move very much, and that means that we don't have to simulate for a long time in order to get reliable answers, because we don't have to wait for things to move. HIV integrase is a dimer, so that's one subunit, that's another one colored differently. These magnesiums here exist near the catalytic site for integration of the proviral DNA. This site here binds to LegF for the translocation of this complex across the nucleus. So if you can inhibit this allosteric site, this LegF site, then you can hopefully like prevent retroviral DNA integration. That's the aim. So the ligands that bind look like this. This allosteric site is very rigid. The molecules are very rigid. So we're going to run some free energy methods and compare them. That's the aim. But before we did that, we wanted to make sure that our molecular dynamics method recapitulated the experimental information on how these things bind. And to do that, we ran an unbiased simulation of 200 nanoseconds to see, and what we would expect is that this ligand does not leave the binding site in 200 nanoseconds, because we know they're low nanomolar inhibitors. We know that by experiment. So they should not leave on the timescales we can access. And they don't, so they just stayed there. They moved a bit. And this is an overlay of the structures over 200 nanoseconds. I think one structure every 20 nanoseconds, and yeah, it didn't move. So the ligand was stable. That's what we'd expect. Our methods are working. So we decided to test for energy methods, and specifically 3 different classes of free energy methods and docking. Well, for 3 classes, but multiple different free energy methods actually. The first class was the endpoint method, and with that method you take the endpoint energy, which is the energy of, or the average energy of the protein ligand complex, and the initial point energy, so the free protein and the free ligand, and you, by subtraction of those energies, you can get the delta G of binding. That's how endpoint methods work. We look at 2 of those endpoint methods, the molecular mechanics Poisson-Boltzmann surface area approximation, and the molecular mechanics generalized Born surface area approximation. So 2 different methods which vary on the type of solvation model used in the approximation. The second method we're looking at are steered simulations, and by steered, it's like you're forcing something, you're steering it towards an endpoint. Instead of just simulating the 2 endpoints, what you do is you start from the bound protein ligand complex and you apply a biasing force to pull the ligand out of the binding site. And because energy is force times distance, obviously, what you can do is calculate this potential of mean force curve. So a potential of mean force curve is the energy required to pull, or the average energy required to pull the ligand out of the binding site at each distance. So you can generate this curve from the average amount of force it requires to pull out, and obviously it should plateau to some maximum force or maximum energy. So you can get the delta G of binding from that. This is the initial state, that's the end state. You can do these simulations as one long pulling simulation, or you can use this enhanced sampling method or enhanced MD simulation method called umbrella sampling. And what that is you have the ligand constrained at different distance increments from the binding site, and you measure the energy required to constrain it at each of those distances, and from each distance you can stitch together the PMF curve at the end, or the potential of mean force curve at the end. So that's 2 different ways of doing that. Arguably the most accurate method, and the method used by scientists at Schrödinger, is free energy perturbation, and there's different mathematical variants of this which differ in how you calculate the delta G of binding from doing this, but the idea is that you have a bound ligand A and a separate bound ligand B, and you transform over progressive simulations the ligand into another ligand. This is relative free energy perturbation. And over those progressive simulations, you can get a difference in free energy of binding of the two ligands. So you get a delta G, the difference in free energy between ligand A and ligand B, just by transforming ligand A into ligand B. That's relative free energy perturbation. Absolute free energy perturbation is when you transform an interacting ligand into dummy atoms that don't interact with the environment. And in that way, you can get an absolute free energy, a delta G. So we're just differentiating between the two: relative free energy perturbation, absolute. And we're going to benchmark each of these three different classes of free energy method. Now, as I said, in the literature, there are comparisons of these methods, but often they don't make as broad a comparison as what we're making here. They'll compare two, as a maximum, three different methods. And from the multiple different studies where they compare two or three different methods, you can get some sort of hierarchy in terms of speed and accuracy for the methods. So molecular docking is not a free energy method, but the scoring function used is meant to approximate the free energy of binding. So we're including that here. And it's obviously the most expedient method, but the accuracy is typically quite poor. Endpoint approximations, as I just described them before, they're a little bit slower, but the accuracy is meant to be marginally improved. The steered molecular dynamics simulations, with or without umbrella sampling, they are meant to be even more accurate still, and the accuracy can be system specific, but the speed is sacrificed again. Free energy perturbation is arguably the most accurate method depending on the system, but again, the speed is compromised. So what we can say from this is that molecular docking and endpoint approximations are probably useful for high throughput screening of large compound libraries against targets, whereas the free energy perturbation and steered methods are probably more useful for, what would you call it? Mental blank. So they're probably more useful for hit to lead sort of scenarios where you want to optimize the binding for energy of specific hits. So you can make small changes and see what happens, but they're obviously slower. So we're going to provide a comparison that is broader than previously reported with, and specific to HIV integrase, because obviously these methods can be system specific. And we're going to provide that comparison here. So what we did was we obviously tested these methods on the protein ligand complexes that I showed you earlier. And from docking, this is the accuracy we got. So on the vertical axis in each case, we have the predicted version of the free energy of binding. So the GlideXP score, as I said, was not a free energy of binding, but it's meant to approximate it. So we're just including it there. The experimental delta G is on, from the paper published by Theta et al. I gave the reference earlier, but the experimental delta G is along the horizontal axis in each case. And from docking, we get an average error. So, well, I should first say that this line here, this black line, is predicted equals experiment. So it's just saying that predicted equals experiment. The green dots, they're plus or minus 4.2 kilojoules per mole, or 1 kilocalorie per mole. And the red dots, they're plus or minus 8.4 kilojoules per mole, or 2 kilocalories per mole from the experimental value. So as you can see from docking, they're consistently outside the red dots. We want our predictions to mainly be inside the red dots, because there was a paper saying that predictions that had errors of 2 kilocalories per mole could still be useful. I can send you the link to that paper. It's a thought experiment where they're saying it's better than going in blind if you have errors of 2 kilocalories per mole. So we want the predictions to be inside the red dots. And as I was starting to say earlier, docking gave an average residual or an RMSE error from experiment of 16 kilojoules per mole. So that was the error we got. And the accuracy of the speed was seconds per ligand. So it was quick, but the error was not what we wanted. In this specific test system, when we used the endpoint approximation MMGBSA, the error was elevated and we saw this spread relative to experiment. The experimental values were meant to be all very close together, but the spread we got was not good. So the RMSE was 25.4 kilojoules per mole, and we got minutes per ligand. So in this specific case, it was slower and the error was still bad. When we used MMPBSA with the methods that we had, we had a marginally reduced spread and an RMSE of 20.7 kilojoules per mole, and the speed was hours per ligand. So that's the kind of accuracy we could expect from this test system. It's pretty rubbish so far. It gets better. So for umbrella sampling, we reduce the spread and we get an RMSE of 18.2 kilojoules per mole in days per ligand. For bar absolute, if we exclude this outlier here, then we get an RMSE of 8.7 kilojoules per mole and a speed of about 1 week per ligand on the cluster in the settings where we're using. So a lot of these predictions here this is for energy perturbation, by the way. Bar is just one variant. So for a lot of these predictions, they are within the red lines, and that's good. For bar relative, where we're transforming between ligands now, so this is a relative delta G. So the difference in free energy between the two ligands, or the difference between binding free energy, was quite accurate. So we get an RMSE of 5.2 kilojoules per mole for 14 relative perturbation, and it was not the slowest method with speeds of days per ligand. We think this spread might be potentially due to force field problems, but because the one important thing that I want to say from these methods is that while we can't really extrapolate these results to different test systems, what we can do is say that for HIV integrase, the only method worth using was bar relative, because they're all within the red lines, and that's the aim we were trying to make. The error is good, and we should, by comparison, we should say that ITC varies itself by 2.1 kilojoules per mole for repeated experiments. So this is quite good. We can argue it's still useful, and for HIV integrase, this is the only method worth using. We made some prospective predictions with the best method. As I said, we're going to leave validation to a later date. These compounds were reported in the paper by Fader et al., but the binding affinity data was not reported. So we've made some predictions where we don't know the answer. They're here. I can send these slides to you later if you want to look over them. So to summarize, bar relative had an error of 5.2 kilojoules per mole from experiment. It was the most accurate method, but not the slowest. And it was the only method worth using for the integrase ligand specifically. We gave 6 prospective predictions where the binding affinity data was not known, and we leave validation to a later date. Case 2. So we're looking at 3 cases. Now I don't need to tell you guys about neuraminidase, but I'll give a brief background anyway for the people joining in. We at MIPS, Monash in Parkville, in Melbourne, we developed a method that works for these specific protein ligand complexes, and we're going to predict PIC50 values from the relative binding free energies that we get. So that's the aim of this. Obviously, neuraminidase is a protein on the viral surface of influenza virus, and Mark Vonigstein and collaborators and his group members, they developed Relenza, and what that does is inhibit neuraminidase. And what neuraminidase does is, as the viral progeny are budding from the viral surface, they are attached to the viral surface by linkage, and the linkage is through a molecule called sialic acid or neuraminic acid. Neuraminidase hydrolyzes. It's a glycohydrolase. It hydrolyzes that neuraminidase acid linkage and freeze the viral progeny. So obviously, if you inhibit neuraminidase, the viral progeny are stuck. And the idea is that the immune system can mop up the virus. And this is a validated mechanism for anti-influenza viral drugs. And as I said, there's compounds discovered by the von Itzstein group that inhibit this molecule, and Relenza is notably one of them. It's a therapeutic that is available. And there's also Tamiflu. So neuraminidase inhibitors block the release of viral progeny, and we're going to take the best method from case 1, which was bar relative, and apply that to ligands binding to the active site of neuraminidase. So tetrameric and N2 neuraminidase looks like this. A lot of previous studies reported where they've done similar stuff to me. What they did was use monomeric N2 neuraminidase, and we find that the stability of the active site requires that we use tetrameric N2 neuraminidase on longer timescales than has previously been reported. As I've said, computers have improved. N2 neuraminidase exists as this tetramer. I've shown here Tamiflu, if you can see it. There's Tamiflu in the active site. And there's a zoom in of the active site here also with Tamiflu bound, and I've overlaid N2 and N9 neuraminidase. So there are obviously multiple subtypes of neuraminidase. The N2 subtype is shown with white ribbons, and the N9 subtype is shown with red ribbons. And here is this 150 loop, and this is in the active site. I've colored it in red. The 150 loop is named the 150 loop because it starts roughly at residue 150. And you can see from the overlay of N2 and N9 that there is some movement in the 150 loop. The 450 loop is up here in black. And underneath the 430 loop, the 430 loop is in black. And underneath the 430 loop is the 430 cavity. There's three important arginines in the active site. The carboxylate group of Relenza and Tamiflu is shown, interacts with these three arginines. And these are key interactions that need to be made and remain stable. And this amine group here on Relenza and the equivalent isosterically replaced guanidinium group of Relenza, these poke into the 150 cavity, and the volume of the 150 cavity will change depending on what group is here, what amine group is here. So what we're going to look at doing is using relative free energy perturbation to transform between different ligands with different substituents here, different unsaturation points, and transform between ligands like this in order to get a relative binding free energy. Obviously, the ligands will look like this. So 5N-acetyl neuraminic acid, or Neu5Ac, is the native substrate of neuraminidase. It looks like this. You can see in red this glycerol side chain. On carbon 3, you can see this hydroxy group. The transition state inhibitor of neuraminidase looks like this. So obviously you just put an alkene here, and that improves binding affinity with neuraminidase. With Relenza, you replace the carbon 3 or the C3 hydroxy with a guanidinium group, and that projects into the 150 cavity, and obviously the volume changes. The equivalent glycerol side chain is shown here in red. Tamiflu carboxylate replaces the guanidinium group with an amine and replaces the glycerol side chain with an isopentyl ether group. The ring oxygen of the pyranose scaffold is removed and replaced with a carbon. So therefore, the positioning of the alkene is different as well. And both of these compounds inhibit neuraminidase. In 2012, the von Itzstein group had this very clever idea of taking glucuronic acid analogues that look like Tamiflu, but contain a pyranose scaffold. And we're going to take ligands based on Tamiflu carboxylate, Relenza-like ligands as well, and we're going to take glucuronic acid ligands in this study and transform between them and hopefully recapitulate the experimental PIC50s that we see published in the literature. These ligands here are going to be the focus of the study because there's a fair few of them published. So one thing I need to emphasize is that IC50 should correlate to KD. They should be proportional. That's true. That's the first point. The second point is that the delta G of binding is a function of KD. So you take the log of KD, multiply it by some constant, and that equals the delta G of binding. That is also true. That's the second fact. Therefore, by extension, the PIC50, which is the negative log of IC50, should correlate to the log of KD, which is, or a function of the log of KD, which is the delta G of binding. So these three things are true. So what you can do is then calculate the delta G of binding by free energy perturbation, and that should correlate to the changes or the equivalent changes in PIC50 or the delta IC50. So here's the data we're using. Here's the PIC50 values for all our compounds. Here are the compounds we have. I can show you the slides later, but obviously the IC50 values or the PIC50 values are ranging from about 10 to about 5.5. So that's about 4 orders of magnitude in log values, right? So it's that should correlate to about, I don't know, you get the idea, about 10,000 in IC50 units. We're going to use relative free energy perturbation to get delta G values, and we're going to use that to correlate to delta PIC50 values, as I said. And here's how relative free energy perturbation works, just as a refresher. You obviously convert the protein ligand complex A into protein ligand complex B with the same protein and get a delta G of binding between those two. And then you take the free ligand in solution and the free ligand B in solution, so the two different ligands in solution, and get a delta G in solution by transforming. And from these two transformations by subtraction, you can get the delta G of binding. This is true because of the closed thermodynamic cycle. I can send some papers later, but that's just a brief introduction. We made several transformations between ligands. So the ligands in all the transformations where the compounds are shown in blue, these are based on the glucuronide structures. And you have the free carboxylate there. And this is what they look like. The ligands here in black are based on Tamiflu. So we're transforming between Tamiflu-like structures and glucuronic acid-based analogs. And we did that using the relative free energy perturbation approach. The numbers in green, so these numbers are corrected. The numbers in red are raw data just by using the relative free energy perturbation approach. So we used, one thing I should stress is that the correction is based on the fact that if you transform from 1B to 6, from 6 to 4, from 4 to 2, and back to 1B, if you go in a cycle all the way back to 1B, the net free energy change should be 0. So if you add up all these red numbers that you get from relative free energy perturbation and the answer is not 0, then you can correct for this cycle not being 0 and all of the cycles not summing up to 0 in the statistically most likely way in the manner proposed by Wang et al. And then you get these corrected green numbers where if you sum -11.9, -9.95 and so on, if you sum all these green numbers, then it adds up to 0. And that is true for every single closed cycle in this perturbation map. So that's what we did. And let's see the results stack up relative to experiment. So we obviously put experimental delta PIC50 values on the vertical axis and predicted delta G values on the horizontal axis. And we put a line of best fit by a simple linear regression through this data. And the correlation we got between the experimental and the predicted value was R equals -0.75 from the raw values. From the line of best fit, the average residual was 1.56 units, or not units, but log units, you know what I mean? So the average error was in the predicted delta PIC50 value from the line of best fit was 1.56, so that's the error. once we do the correction, oh, I should also say these dotted lines, this is the 95% confidence interval in the line of best fit. So there's 1, 2, 3, 4, 5 little dots outside this confidence interval. Once we do the correction, once we use this hysteresis correction algorithm, everything improves so much. And we're not doing this with knowledge of the experimental information. The correlation to experiment just improves by virtue of the fact that we're doing that correction I mentioned. The R value between the experimental value and the predicted value goes up to negative 0.96. That's an R squared of 0.92. So 92% of the variance in the delta PIC50 values from experiment is explained by the predicted delta delta G values. That's quite good. And this R value is obviously significant. Importantly, the average residual from the line of best fit was 0.62. So taking a predicted experimental delta PIC50 value from the predicted delta delta G value, you get an error of 0.62. By comparison, so with Tamiflu carboxylate, plus or minus 2 standard deviation from the replicate of 3 experiments in the assay, 3 replicate experiments, that standard deviation from that in PIC50 units was, or 2 standard deviation, sorry, was 0.46. So that's the kind of error we would expect from replicate assays. This is the kind of error we're getting from the modeling. Importantly, this modeling can be done more quickly than the assay, so it can be used as a guide. That's the idea. We put error bars in here as well. And oh yeah, the 95% confidence interval in the prediction goes down. So 1, 2, 3, 4, I think maybe 4 things are outside the 95% confidence interval in the line of best fit. So obviously the correction improved things and our accuracy is approaching experiment. That's what we would contend. So we have clarified the best MD parameters to use for N2 neuraminidase inhibitors. Previous studies have used the monomer. We've shown that the active site 150 loop does not remain stable on longer timescales than previously accessed if you use the monomer. And that's just, we found that by virtue of the fact that computers are improving. We found that this specific force field, this specific list of MD parameters or molecular dynamics parameters was required out of everything we tried. So there might be other combinations that work, but we didn't try them. Out of the extensive list of force fields that we tried, the AMPA 99 SB force field was the best. The GAF force field was required for ligands, and the TIP4P water model was required for bound water molecules in the active site. We used the best method from our comparative study, and we predict the delta delta G of binding. Correlations between predicted and experimental values were excellent. So we got an R squared of 0.92, I think, after the hysteresis correction. And our accuracy is slowly approaching the accuracy of experiment. So as computers improve, we'd expect this also to improve. Where I was originally going with this work was that I hope to use these methods for homologous glycohydrolases to neuraminidase. So for example, the human para-influenza virus hemagglutinin neuraminidase, we could use that as a dimer and run equivalent calculations. The putative pseudominidase, or the pseudominic acid carbohydrolase, that was reported by Taylor et al. In 2008 and detailed in reviews by the Kiefel group. We could apply similar methods to that. This exists natively as a trimer. Other 8 or 9 carbon acidic sugar carbohydrolases resembling a neuraminidase, as they're discovered, we could obviously apply these same methods. All right, I'm really excited about this, and I've got 10 minutes left to present it. So this is the work, and I noticed that my supervisor is in the chat, so I'm really excited to present this. We wanted to help with the current pandemic. That was our main driving motivation. And we applied some of these methods to see whether we could actually make some useful predictions with computers. So we published this paper in JPhys Chem B, and we're looking at whether an active site inhibitor induces conformational penalties for the ACE2 recognition by the spike protein of SARS-CoV-2. So that's what we're looking at. And obviously for SARS-CoV-2, the virus will bind to the host cell. It's another retrovirus. It binds or adheres to the cell surface via interactions between the spike protein and cell surface bound ACE2. So our idea was if we could change the conformation of ACE2 by binding an active site inhibitor, will that perturb in any way, in any measurable way, the binding of the viral spike protein? It was sort of an academic exercise, and the idea was if we saw any reduction, this could give hope that future inhibitors could further reduce the binding of SARS-CoV-2 to the cell. So that's the idea. And we're looking at this key point here. So some important things. We can define the SARS-CoV-2 S protein binding site in terms of these blue protein loops here, and combined with the red protein loop. So it's the N-terminal alpha-1 and alpha-2 helices starting at serine 19, and also the protein loop going from residues 325 to 355. And the active site of ACE2 is obviously in here. This loop in red can be seen to be somewhat near the active site. Importantly, when an active site inhibitor binds, we see by crystallography that there is like a hinge motion. ACE2 folds inward to cover the bound active site inhibitor. And we wanted to know if this hinge motion, this change in conformation that we see, would perturb the binding of SARS-CoV-2 S protein. That's what we wanted to see. So our methods, we built the systems. We built system 1, ApoACE2, system 2, the inhibitor MLN4760 bound to ACE2, system 3, the ACE2 S protein complex, and system 4, the MLN4760 ACE2 S protein complex. We built that system. We equilibrated that system and ran MD simulations. So we heated it up, we applied pressure into an MBT ensemble, and we ran 1.5 microsecond MD simulations in triplicate with randomized initial velocities for each simulation. And we aimed to analyze these trajectories. So we inferred the delta S, the entropy of binding, from looking at the changes in the ensemble average RMSD and the protein motifs. So if ACE2 changed its variation or fluctuation in RMSD upon binding of the active site inhibitor, then that would obviously be an inference that there is a change of entropy upon S protein binding, upon MLN4760 binding, and so on. We inferred delta H by monitoring the key interactions between ACE2 and the S protein. And we also ran an endpoint calculation, MMPBSA, to calculate a general idea of what the change in system enthalpy should be. So we ran our MD simulations, and I've got a video here. In orange is the S protein, in green is ACE2, and you can see the bound active site inhibitor there. Importantly, the crystal structure coordinates, or cryo-EM coordinates for this complex did not exist. So we built it as a hybrid of the hollow MLN ACE2 crystal structure and the crystal structure of ApoACE2 and the S protein bound. So those two crystal structures exist, and by hybridizing the coordinates, we were able to generate this structure, and we ran it for a combined 1.8 microseconds in total. We found dynamic residues not previously reported by MD. So these residues here in purple, these changed by 8.2 angstroms RMSD relative to ApoACE2 during MD simulation. And there's all these other key changes. So the S protein binding site changed by 5.5 angstroms RMSD relative to ApoACE2 when the active site inhibitor was bound. And we wanted to know whether these large changes in conformation would affect the binding of S protein. And this could help with the design of future potential inhibitors. So what we saw was that when MLN 4760 was bound, the interactions from ACE2 to the S protein involving ACE2 residues, glutamate 35, glutamate 37, tyrosine 83, lysine 353, and arginine 353, these interactions with the S protein were weakened by more than 10%. So the bond occupancy of these interactions was weakened by more than 10%. Interestingly, the ACE2 GLN24 interaction was stabilized, and perhaps future potential inhibitors could target this interaction. So we propose that the change in bond occupancies is likely due to the change in the scaffold architecture when MLN 4760 was bound. So we ran MMPBC calculations first of system 3 and then of system 4. So we looked at ApoACE2 bound to the S protein and also holoACE2 with MLN 4760 bound to the S protein. And we found that the enthalpy of bounding S protein binding was marginally reduced when MLN 4760 is bound. That's what we found, and we propose this is likely due to the changed geometry of the S protein binding site. I'll just rush through. We can do a per residue energy decomposition. So we found that these specific residues contributed to an enthalpic benefit of 10 kilojoules per mole or more in both systems. And these residues in bold here, so glutamine 24, so they're all shown there. They're the ones I'm talking about. And also, as you're looking through this graph, like in blue, you see system 3, ACE2S protein. In red, you see system 4. And there is a marginal reduction in the enthalpy as you go from system 3 to system 4. So that's what we saw. So some key points. We found by MMPBSA analysis that there is a 1.48-fold reduction in the delta H, or the enthalpy of binding, when MLN 4760 is bound, and this should correlate to some sort of equivalent experimental value. And this is the upper limit of a 95% confidence interval, because these simulations were run in triplicate. And we propose that this is likely due to the changed geometry of the S protein binding site. These specific interactions reduced by 10%. These specific interactions, so they reduced by 10% when MLN 4760 is bound. These specific interactions here contribute to an enthalpic benefit of 10 kilojoules per mole or more when S protein, in both systems. So we could target these residues in the design of future inhibitors. We intend to do future work with future inhibitors of ACE2 to see if they induce different conformations to ACE2 and further reduce the S protein binding. So I want to thank everyone here. David Chalmers, I noticed Elizabeth was in the chat. I haven't checked, but I'm really thankful for David as well for helping me at MIPS. I'd like to thank the Integraze group specifically, the SBSB group and the Scanlon group, everyone named here, as well as Monash University in general, the NCI, Massive, VLSCI for making my PhD material possible. And also Irene for taking a risk with me and hiring me as a new postdoc. Meeble, so the whole COVID idea, the whole COVID project was her idea, and without her it wouldn't be possible. Everyone in this photo here, that's the team at RMIT. I'd like to thank the Australian government, the ARC, for basically paying me, NCI and Spartan. So these computing infrastructures for providing invaluable computational resources. This link here is to the modeling group at RMIT. So here's everyone: Matt, Tu, Wen, Irene, she's amazing. Ben, Irene, Nev for helping me, Pat, and there's me in the corner there just as I started. I'd like to thank everyone for my personal life. Yeah, so I don't know if we have time, but questions?
Excellent. Thanks, Billy. I'm just going to see if I can, how do I do this? Hang on. I was trying to get you up on the screen here, Billy, but I think you're there.
Would you like me to, I could just stop sharing and then, does that work?
That might work. All right, so the chat's there. If people want to ask questions through the chat, they can. If people here in the audience want to ask questions, I have to then translate it. I don't know if I've got that online. So does anyone in the audience here have a question? Can you see the chat, Billy?
Yeah, I can. I'll just.
Do you want to address that?
Yeah. So from Yunshi, he says, thank you for the talk. What do you think is the current bottleneck for improving accuracy, force field quality or computational power? Oh, that's an interesting question. I think both, personally. I think if we used polarizable force fields, then that should improve things. So for those who don't know, force fields are a list of parameters that tell atoms how to do things during MD parameters. So if we obviously had more complex and detailed force fields that more accurately modeled reality, then that would improve accuracy. Also, we need better computers as well, and I'm excited to see what the future brings. So as computers get better, we can obviously use more complicated methods. Question. RE distribution based on hysteresis of up to 20 kilojoules per mole and 2 dataset points to a bigger problem. The closed thermodynamic cycle delta delta G not adding up to 0 points to other likely problems, force field for ligands incorrect binding mode, individual free energy calculations not converged. Error bar, have you investigated any of this? So the hysteresis of up to 20 kilojoules per mole. Let me just open up the thermodynamic cycle. So I'm gonna share my screen again. So with the hysteresis here, so here. Yeah, I see what you mean. At certain edges, there's hysteresis, 15 there, 7 there. There's not often hysteresis of 15 kilojoules per mole, but I agree there were significant challenges in selecting the force field for neuraminidase modeling. One problem was that if you use the incorrect force field, then the tri-arginal cluster would separate. So each of those arginines is obviously positive. So force field choice was important for not causing those arginines to repel in a non-physical way that you don't see by experiment. Yeah, this one edge is obviously problematic. We could probably look at running another couple simulations or looking at a different binding mode or equilibrating for longer before running the free energy calculation. But no, we didn't look at that, Alpesh. It was quite struggling to run tetrameric neuraminidase because that system was very large. During my PhD, I don't think we got a lot of time to look at this one specific edge. That one there, 7 is not too bad. I don't think it's 1.5 kilocalories, 1 and 1.7. Yeah, we haven't looked at it too much, but importantly, we found a force field that seems to work now. I have to stop sharing the screen to see new messages.
I think there's a question from Todd.
Nice talk, Billy. Would ACE2 inhibitors be most useful pre-infection? So we're not showing that the inhibitors work yet. So the inhibitors we looked would not be viable therapeutics. But what we have shown is that there is a marginal reduction in binding strength. And that marginal reduction in binding strength might not correlate to a reduction in infectivity by assay. But what we would propose is that future inhibitors might change the ACE2 binding architecture in a different way so as to be better as possible therapeutics. We wouldn't suggest that these are potential therapeutics at all. What we would suggest is that as an academic exercise, we can show that binding of an ACE2 active site inhibitor does marginally reduce binding strength.
Excellent, thanks, Billy. Any questions from the audience here? Thomas Hasselhorst has a question for you, Billy.
Billy, can you hear me?
Yep.
Yeah,
Speak louder.
So the effect on S protein binding to ACE2 in the presence of the ACE2 inhibitor. Look, have done the SBR experiment in the presence of ML4760, and it does really not to a great extent change the affinity to the spike protein. So I believe that what you have shown, there is a change, but does it really matter in the sense of the overall viral receptor interaction?
Thank you so much for the question. I'm struggling to hear. Milton, I missed most of that.
I'll paraphrase it, Billy. I think what Thomas was saying was that that experiments have been done here where they've looked at the S protein binding with an inhibitor bound, the same inhibitor, I think, was it the ML?
Yeah,
similar. Yeah, into the ACE2 active site, and it really has no, having an inhibitor bound really has no impact on the ability of the S protein to bind. But I think your experiments are quite interesting there, Billy. Anyway, so.
Well, yeah, we saw a marginal reduction. It was very small. Was it by SBR or by ITC?
SPR.
SBR. Yeah, it might depend on the construct of the S protein used. It might depend on a lot of things, but we're not suggesting there would be even a noticeable reduction. By our experiments, we would suggest that there's a very small reduction. But it's interesting you say that there is no observable reduction seen by experiment. That could be interesting for future work. We've got some other results that are confidential, and they might suggest toward the same thing, actually. So it's interesting you've pointed that out.
All right. With one eye on the clock, Billy, I'm just going to ask a very quick last question.
Yep.
You mentioned really early on in your talk about how, ultimately the aim here is to see if computers can predict comparable to doing the experimental work and actually measuring the numbers. So based on what you've presented, maybe we're not quite there. So I just wondered, how long do you think it will be before we do get there? So where you can crunch numbers and go, we guarantee that if you make this compound, it'll work.
I think it's a good question. I think if I wanted to put a time frame on it, maybe in the next 50 years at least. But I don't know. It would take. But importantly, we're not saying that we're as accurate as experiment. What we're saying is that we can guide experiment.
Yep.
So the weatherman makes predictions about the weather, and you'd be silly if you thought he was right all the time. But still, people rely on him. So in the absence of like experiments that can give you the information you want, maybe molecular dynamics could be used as a useful guide to complement experiment at the moment. And there's heaps of examples where that's been done and where it's useful. And I don't think the predictions made by MD are totally unreliable. I think they do approach experiment, and there is definite room for improvement. I think there was a question about how could we improve the accuracy and what's causing the current bottleneck. And I think it's to do with the way we run calculations at the moment. Obviously, if we use the most sophisticated quantum mechanical calculations over a large enough time, then we'd get very accurate results. But there's a bottleneck due to the way we run calculations at the moment, and to improve how we do things, we'd need better computers. But no, computers are a useful guide at the moment. And I think that analogy with the weatherman kind of works. You still believe him when he says it's going to be sunny the day before, and you might have an 80% chance of being right. And I think some sort of margin of error is acceptable.
Yep. Good. Hey, thanks, Billy. Look, it's been great to hear what you've been doing for the last few years, Billy, from my personal point of view. It's a pity you couldn't get here, but look, we among many other things, we don't control the weather, and we don't control COVID outbreaks either. So,
well, I'm glad I can make the background.
Safe in Melbourne.
Yeah, you guys too, and thank you so much for having me. Thank you for the organisers as well. I really appreciate you having me here.
You can get up here at some point, Billy, and we can catch up.
Yeah, I look forward to it. I'd like to make time. Great. I'll let you know.
Okay, thanks, Billy. All the best.
Thank you, Milton. Thank you, everyone.